Infectious diseases have been the scourge of mankind probably long before the advent of recorded history. Infectious diseases are those caused by pathogens. Some of these pathogens are capable of jumping from species to species, and in the case of pathogens that infect both man and animals, these are called zoonotic diseases. Most of these diseases are caused by parasitic, bacterial, or viral pathogens that are easily spread from host to host, either directly or through vectors such as mosquitoes or biting flies. Before we understood infectious diseases, their causes, and how they spread, outbreaks of widespread illness and death of epidemic proportions exacted dreadful losses of well-being and even life upon mankind. Pathogens have had a major impact on society throughout history, from biblical times with leprosy to medieval times with the Black Death, the settling of North America by Europeans with smallpox, the building of the Panama Canal with yellow fever, the 1918 influenza pandemic, and even in current times with Ebola, just to name a few. Vaccines can prevent many of these diseases, while antibiotics and antiviral drugs can be used for bacterial and some viral infections. Nonetheless, tuberculosis and malaria continue as major killers due to lack of effective means for prevention and/or treatment. Viruses enter the body and may encounter an immune system's first line of defense, the macrophage. If the virus is engulfed by the macrophage, it may get destroyed, and bits and pieces of the virus may be used by the macrophage to activate the immune system. It takes time to develop a new specific immune response against a new pathogen when the immune system encounters it for the first time. Specific immune responses occur through the coordination of activating cells like the macrophage or dendritic cells that monitor for pathogens. Lymphocytes are immune cells capable of highly discriminating specific responses against pathogens. B lymphocytes make antibodies which circulate in the blood and can directly bind to the virus and cause its neutralization. Macrophages or dendritic cells activate T lymphocytes, which, depending on type, either help B lymphocytes make antibodies, activate macrophages, or develop into cytotoxic T cells that specifically recognize and kill pathogen-infected cells. These pathogen-specific lymphocyte responses are highly controlled to attack only the pathogen and not normal tissues or cells. Once that pathogen has been eliminated, the activated B and T lymphocytes become much less active, and some become immune memory lymphocytes. The memory lymphocytes are in a resting state that allows them to be more easily activated. These memory lymphocytes develop to provide a more rapid and potent immunity response should the body be invaded by the pathogen again. Vaccines work by pre-stimulating the immune system so it is ready with antibodies and memory B and T lymphocytes to respond more rapidly and strongly should a particular pathogen invade the body again. The concept of herd immunity with respect to vaccination is important to prevent the spread of pathogens throughout populations. The higher the proportion of immune individuals, the fewer susceptible ones, and spread throughout a population can be stopped if there are enough immune subjects. For example, with only 50% of a group being immune, there remains half of the group that can become infected and spread the pathogens to others. However, with 95% coverage of immune individuals, susceptible ones are rare and are actually protected from infection by the 95% who are immune. Welcome to BioMARC. We are part of the Infectious Disease Research Center at CSU. BioMARC is a contract development and manufacturing organization that provides research and development services to move products that require high biocontaminant to eventual commercialization. We specialize in vaccines, diagnostics, and biotherapeutics produced under GMP compliance. We serve private and government clients in the biotechnology and pharmaceutical sectors. BioMARC manufacturing is under strict regulation and guidance from the FDA. This requires stringent controls to ensure patient safety and product purity and potency. Production laboratories have specific clean room requirements to ensure processes are as clean as possible. This includes the wearing of special Tyvek suits with gloves to reduce unwanted particles, environmental monitoring in production rooms, and meticulous attention to see what process procedures are specifically followed to the letter. Manufacturing rooms are constructed of solid surfaces with sealed light fixtures and utility ports to facilitate cleaning and to reduce environmental contaminants such as pathogens and dust particles, and to enable processes to be conducted in areas that meet FDA-required specifications for biopharmaceutical manufacturing. Airflow for environmental control is carefully monitored and adjusted through building automation systems to ensure high biocontaminant of pathogens in rooms where organisms are grown through a constant inflow of air. However, for other operations such as fractionations, purifications, and final product formulation and filling into vials, the airflow is controlled to have a constant outflow to reduce incursion of unwanted organisms and particles into these downstream processes. Manufacturing equipment is highly specialized and often of a single purpose or dedicated to a particular production process. In this example, a cell, representing one of perhaps hundreds of millions of cells in the bioreactor, is internally filling with replicating virus to the point the cell ruptures and releases the virus into the culture medium. Some viruses don't freely flow from the infected cell, and additional steps are needed to break the virus free of the cell. The next steps involve the harvest of the virus in the culture medium and eventual purification. As can be seen in this case, the cell ruptures, releasing not only the virus, but also the structural and internal components of the cell, including plasma membrane fragments, enzymes, proteins, and nucleic acids, all of which represent contaminants for the virus preparation. Various strategies and techniques are used to separate contaminants from the target. These commonly include filtration and chromatography, which are manufacturing friendly and which can result in highly purified virus preparations. Eventually, the purified virus is formulated into a vaccine with biocompatible materials that may enhance the immune response, as well as enhance the stability of the vaccine under storage and field conditions, depending on what those may be and wherever the vaccine is shipped for use. Quality control is a critical aspect of biopharmaceutical manufacturing. At various stages during the manufacturing process, samples are analyzed to ensure production proceeds as expected to enable manufacture of the final drug product or vaccine. Molecular, biological, and biochemical analytical procedures are used as in-process monitoring controls or for final product release. Testing by the Quality Control Department is reviewed and certified by the Quality Assurance Department to ensure analytical accuracy and compliance with established standards. Careful quality control and quality assurance measures ensure patient safety and product quality and potency. BioMARC emphasizes focus on infectious disease solutions that target unmet needs. Of particular importance are emerging diseases with potential for global spread, like Ebola, Zika, or Rift Valley fever, which have origins in lower and middle income nations. Such areas have challenges such as less developed infrastructure, healthcare system access, geopolitical unrest, and restricted economic opportunities. Vaccine and diagnostic solutions may meet needs in endemic areas, but may also be important to areas threatened by global expansion such as the United States. Whether the project be a biodefense project or one that serves the greater good, we at BioMARC drive to make impact and a difference in the fight against infectious diseases.